Okay guys, so last time we ended up finishing off with our protein structure. We went over our 4 levels of protein folding, and then we finished up going over collagen. So today we're going to start off by going over red blood cells and hemoglobin, which is a protein on our red blood cells. And what this does is it's an oxygen binding protein. So the nice thing about this is as the red blood cells move through our body, they can carry the oxygen with them through this protein called hemoglobin. And hemoglobin has 4 polypeptides that make it up. So along with our red blood cells, there is this disease called sickle cell disease. And with sickle cell, we are going to have, and I've actually had students in the past that have had sickle cell disease, and they've missed a lot of school whenever this condition, acts up. But with sickle cell disease, the proteins are not folded with these red blood cells the same way they are with a normal red blood cell. So a normal red blood cell is going to be very circular, and it has a lot of surface area in order to bind our hemoglobin, which means it can carry a lot of oxygen. People who have sickle cell disease, a little bit different. Their red blood cells are actually in a sickle shape. And what I mean by this is they look something, oops, redraw that, guys. There we go. They look something like this, as opposed to a normal red blood cell looking somewhat circular. Best of my circular abilities there. All right, so you guys can see a lot less area to bind oxygen. What are the effects of this? Well, if you can't bind oxygen, that's going to be less oxygen going to your muscles, being transported through the blood to wherever it needs to go in your body. And whenever we are going to be using this oxygen later on, it's going to be to make energy. So less oxygen, less energy in the long run. And here's 2 pictures, guys. You guys can see the difference between your normal red blood cell and your sickled one down here. What causes this, guys, is it's 1 amino acid difference on the side here. You guys can see number 6, instead of being GOU, it's VAL, and that 1 little different amino acid there causes the protein to fold completely differently, and that's why we have a different shape in the end. Okay, so let's talk about proteins becoming denatured. So things like pH, salt concentration, temperature, other things like that, they can cause a protein to actually lose its shape and become maybe less active or even completely inactive. And this is called denaturation. So here's a good picture of it. Let me draw you a different one that'll give you a little bit of a better representation of it. So if here's our original enzyme, okay, there's our original enzyme. Let me draw a substrate in there for us. Okay. All right, so here's our enzyme, here's our substrate, and you guys can see that the active site on the enzyme will fit perfectly into the substrate. Now, temperature changes too much, pH changes too much, salt concentration changes too much. Our enzyme is no longer going to look very nice like this and have its very shapely active site. What's going to happen here is our enzyme might turn into something like this. So now here's our new enzyme. It has become denatured. Okay. So what this means is if our substrate here tries to bind to our enzyme, it cannot bind. Okay, so no binding occurs. And if we go back to our graphs from earlier, so if we are going to use an enzyme, if you look at the size of the hill there, it might take about that much energy. If we don't use an enzyme, it might take that much energy, okay? So you guys can see the difference here. If we have a denatured enzyme and we can't use it, like when we're sick, our bodies are going to need a lot more energy to just keep you running. That's why when you're sick, you're not going to feel good. You're going to want to stay in bed the whole day because your enzymes aren't working properly. In other words, your body's using a lot more energy compared to what it normally does, and you have less energy for other activities, I guess you could say. Okay, so what they've tried to do, guys, is determine what a protein looks like, how it folds. In other words, they're going from primary to quaternary sequence and trying to see, okay, hey, what's the amino acid sequence? All right, since they are there, which one's going to fold on which, and which R groups are going to be attracted to others. And they use X-ray crystallography here. You guys can see we have down here RNA polymerase 2. It's one of our RNA polymerases in our body. And what it does is it helps to add our RNA nucleotides to our new RNA molecule. All right. What's nice is our DNA comes through here and it fits very nicely into RNA polymerase 2. So because that RNA polymerase, which is a protein, folds just like that, our DNA can fit perfectly in there. So it's kind of like structure function kind of thing. Because of the structure of RNA polymerase, our DNA can come in, bind, and then RNA polymerase can do its job. All right, last macromolecule. So we already went over carbohydrates, proteins, lipids, and now we got to get to nucleic acids, and that'll be our last one. So two main, I guess we can call them polymers of nucleic acids, are going to be DNA and RNA. So here is a cell, eukaryotic cell. Here's our nucleus. So what happens here, guys, is with respect to our nucleic acids, we have DNA, and then I know we already went over this in the past. We have our DNA. It's getting converted into mRNA because we never want that DNA to leave the nucleus. If you remember, we talked about that. So it's going to take its message, convert it into mRNA. The mRNA can then leave the nucleus. All right, it leaves. It's free to swim about the cytoplasm and find itself a ribosome. When it finds a ribosome, it's going to, and we'll go through this in a later chapter, but it's going to go through the ribosome and it's going to get read. And in the mRNA being read by the ribosome, it's going to get converted into amino acids. And like I said, guys, we'll go over that in a later chapter, how that process works. But this is a basic overview of that. All right, let me fill out the rest of our chart here. Okay, so nucleic acids. I'm going to do the function, the monomer, and the polymer first. And then we will come back to our structure. Oops. Okay. All right, so for our function, guys, pretty easy. Store genetic information. Okay, so there is our function. That was smart of me to put it there, right? Okay, so there's your function, guys. Let me do monomers and polymers. Our monomer is called a nucleotide. There are 4 of them. There's 4 of them for DNA. Adenine, guanine, cytosine, and thymine. That's for DNA. There's also 4 of them for RNA. Still A, still G, still C. Only difference, instead of thymine, it's going to be uracil for RNA. Okay? And then our polymer, it's called polynucleotides. Your big examples, real easy, guys. DNA and RNA. Okay? So there's your monomers and your polymers of nucleic acids. And then lastly, let me do the structure over here. So there's going to be 3 parts of our structure of a nucleotide. We're going to first have, and I'll make these all different colors, guys. We're going to have a phosphate group. Okay. We are then going to have, I'm going to draw all the bonds in red, guys. Okay, so there's our bond. And we're going to have I'm drawing this to the best of my ability. Okay. So for this one, guys, we are going to have a 5-carbon sugar. That's our 5-carbon sugar. Now, this looks probably a little bit different than we went over it in regular biology. Getting a little more advanced here. So if I go around our kind of pentagon thing, this is an oxygen here. All these other points are going to be carbons. So we have carbon 1, carbon 2, carbon 3, carbon 4, and then up here is carbon 5. Okay, that's going to be important later on down the road. Okay. All right, and then lastly, we are going to have another bond here. And right here is going to be either our nitrogenous or nitrogen base, however you want to say it. I'm just going to write nitrogen base. Okay. And that's going to determine if it's going to be adenine, guanine, cytosine, thymine, that kind of thing. Let me write our 5-carbon sugar up here again. So our 5-carbon sugar, guys, is going to be different depending on if it's DNA or RNA. If it is DNA, it's going to be deoxyribose. If it's RNA, it's going to be ribose. So there is a difference between the 5-carbon sugar and the makeup. Not much of a difference, but enough to make it a little different. So, all right. So there is your rest of that chart, guys. So we are done with that one. This slide, pretty much the same thing we just went over. We got our polymers on there, polynucleotides, DNA, RNA, that type of thing. They're made up of nucleotides, and we went over the different parts here. Now, we're going to go over the difference between a nucleotide and a nucleoside. So right here, guys, we have our nitrogen base, nitrogenous base, however you want to say it, our 5-carbon sugar, and our phosphate group. If we are just talking about the sugar and the base, it's called a nucleoside. When we add all 3 of these together, so we've got our phosphate, our sugar, and our nitrogenous base, now it's called a nucleotide. So a little different there. We already went over these guys, nitrogenous bases. All right, we have cytosine, thymine, uracil, adenine, and guanine. The only one that is only in RNA is the uracil. The only one that is only in DNA is thymine. So cytosine, adenine, and guanine are shared by both DNA and RNA. Another one, guys, okay, the DNA and RNA. DNA has deoxyribose as its 5-carbon sugar. RNA has ribose. Okay. So we're going to talk about these 3 and 5 prime, and that's going to be respect to the sugars. So the carbons on the sugar, I went around and I showed you it's 1, 2, 3, 4, and then 5 was up top. Okay, that's going to be very important for us with knowing where things are going to be connecting and what orientation our DNA is in. So we'll go over that in a few minutes, guys. we're usually going to go 5' to 3'. And then one side's going to be 5' to 3', and the other side's going to be 3' to 5'. And I'll do my best in drawing that for you guys to make everything pretty legible. So let me draw that real quick here. All right, I'm only going to draw a few guys because this will take a little bit of time, and I don't want to go too crazy here, and I'm not an artist. So here's our phosphate group. Okay. Attached to that is going to be our 5-carbon sugar. Okay. And then attached to that guy in the middle here is going to be our nitrogen base. Now for this one, I'm going to call it our nitrogen base. We're going to say this is adenine. Okay. Now what we're going to do is we're going to attach another nucleotide to this. All right. All right, so next nucleotide here. And we're going to attach our next nitrogen base. And we'll make that one a T. Okay. I'll draw one more, and then we'll be done. With this side, at least. And we'll make this one, I don't know. Make it a G. Cool. Okay. So, on this one, guys, I'm only going to label like one carbon here. All right, so we have carbon 1, or one sugar, 2, 3, 4, and 5. If you notice, our 5 carbon, we call these prime, our 5' carbons, we have 1', 2', 3', 4' bracket. Our 5' carbon is attached to our phosphate group of that nucleotide. Okay, so this nucleotide is composed of our phosphate here, our 5-carbon sugar, and our nitrogen base. If you look down here, our 3' carbon is going to be attached to our next nucleotide. Okay, so it's going to allow us to attach that next phosphate group, which is then again our next nucleotide. All right, so in this orientation, guys, we say that this top side here is going to be our 5' side. Okay, at the bottom here, this is going to be our 3' side. Okay, now why is all this important? Well, DNA is anti-parallel. What this means is that we are going to have another side of DNA, and it's going to be the opposite of the side we just made. Okay, so this one, we had our G on this side. On the other side, it's going to be attached to a C. A always bonds to T, G always bonds to C. Think of it this way, guys. Stupid little way to remember it. Apple in the tree, car in the garage. Okay, apple, tree, A-T. Apple in the tree, A goes to T. Car in the garage, okay, C and G, they always go together, but yeah. Okay, so again, guys, I'm going to do my best to draw this one. This is where things get a little more difficult. All right. So if you notice here how I drew this differently, we now have our 5-carbon sugar upside down. Okay. We're still going to have, if I put my carbons in, this is carbon 1, 2, 3, 4, 5. Okay. And then this one's going to connect to our next phosphate here. Okay. Bring our carbon. And A would go to T, so let's make our A in there. All right. And let's make our last one. And then we will be done. This side is much harder to draw than the other side, guys. I always have trouble drawing this side. It's like doing everything while we did, but backward. Okay. And then we'd have to draw a T connecting to our A. Okay. Now, if you look here, if we were to label all of our carbons again on this last one, 1, 2, 3, 4, and 5, the third carbon is always pointing this way. So we're going to have this side is going to be 3 prime, and this side is going to be 5 prime down here because the 5 prime carbon is pointing upward. So if you notice, this is why we call DNA anti-parallel. If this is a 5 prime side, across from it, it has to be a 3 prime side. And vice versa, if this is 3 prime, across from it, we have to have a 5 prime. We can never have 5 prime going to 5 prime. It does not work. We also can never have 3 prime going to 3 prime. Okay? So, not good. So we always want 5 prime going to 3 prime, and then the same across from it. All right, guys. Big picture. There we go. Okay, we already talked about this one. Apple in the tree, car in the garage. A always goes to T. G always goes to C. If we are pairing DNA to RNA, it's still going to be the same. A is always going to go to T for, DNA to RNA. Only difference with RNA, if A were to go to T, well, there's no T there. So A is going to go to U. Same thing vice versa. If we have a U in RNA, a U would go to A. All right, here's a short video, guys.
If you imagine your DNA right now, you'd probably picture that double helix you learned about in school at some point, hopefully, but it may actually look a lot different. Hey guys, I'm Sapna here at D-News. That ladder twisted on itself with bonds between 4 bases, adenine, guanine, thymine, and cytosine. That is the DNA double helix, the thing you were probably picturing in your head. But it's now thought DNA at times may have different configurations. The helix can twist the other way, have a couple extra strands. It can even twist onto itself into some pretty cool shapes. There are 3 biologically active types of the double helix: A, B, and Z. B DNA is that recognizable one. It has a right-handed twist, and it's the most common in living cells. A DNA has a wider spiral, and Z DNA twists the other way. It's a left-handed double helix with a zigzag structure. The biological purpose of Z DNA isn't quite clear. Since it twists the other way, it's thought to relieve some of the torsional stress on the structure. And some research shows it may play a role in transcription, where the genetic info is being copied. Beyond the double helix, new research says DNA molecules may also coil into a range of cool shapes called supercoils. Research published in the journal Nature Communications last October shows images of supercoiled DNA created by cryo-electron tomography together with supercomputer simulations. The scientists wanted to know how that DNA crams itself into a tiny nucleus. So they tried to look at it one twist and turn at a time. They found sharp bends, figure eights, even coils that look like handcuffs. Keep in mind this research, like most of the research on DNA, is in vitro or in the laboratory. But by using an enzyme called human topoisomerase 2 alpha, they did try to simulate conditions in the human body to show that supercoiled DNA is possible. So your DNA may be backwards or zigzagged or even twisted, but it might also have double the strands. Most DNA has 2 strands, but many people don't know it can also have 4. It's called G quadruplex DNA or G4 DNA. The G refers to guanine, one of those 4 bases that holds the DNA together and spells out our genetic code. G4 DNA is a topic that scientists are trying to learn more about, especially in the last decade, because it may hold the key to some major medical breakthroughs. Researchers in Sweden recently discovered G4 DNA in one species of yeast. They published their findings in the journal Nucleic Acids Research. Meanwhile, a team led by Florida State researchers looked for and found G4 DNA in the genome of the maize or corn plant. And in 2012, Cambridge University researchers claimed they saw G4 DNA in human cells. Using a fluorescence biomarker, they tagged an antibody they created to specifically look for the 4-strand DNA in human cells and bind to it. They found that G4 DNA seems to turn up most often during the S phase, just before cells are about to divide. Which brings us to the point of why we should care about 4-stranded quadruplex G4 DNA. If it plays a role in cell division, either speeding it up or slowing it down, scientists think G4 DNA may play a role in causing cancer and degenerative diseases in humans. So how do you turn it on or off? And is it possible to create a drug that targets 4-strand DNA and maybe stop cancer? We're not there yet, but that is actually a goal and focus for ongoing research. By the way, have you ever heard someone say, "I love to travel, it's in my DNA"? Is that true? Are some of us hardwired for wanderlust? Watch this video by Julie.
Okay, we're not going to worry about that one, guys. I just thought this was a cool video because it shows us that, yeah, we don't know everything we think we know about DNA right now. And we're still finding out new things. So, yeah. All right. Anyway, guys, so this one, just so you guys know, whenever we are combining our DNA, so we got our nucleotides attached to other nucleotides. When we're talking about one strand of DNA, it's going to be all covalent bonds, okay? So like on the kind of left side here, our 5' down here to our 3' DNA, that's going to be connected by covalent bonds from our nitrogenous base to our 5-carbon sugar to our phosphate group and then to the next nucleotide. But as our 2 different strands of DNA, as our double helix connects, those 2 strands connect to one another, these in the middle here are going to be hydrogen bonds. Okay, so not as strong as our covalent bonds. This is really good later on because we can easily break our DNA in half rather than breaking off single nucleotides on the sides. Okay, we're not going to worry about this one, guys. So here is a real quick chart just on amino acid sequences in different animals, and they were comparing them. Okay, moral of the story, guys, is the less amino acid differences we have in another organism. Okay, so again, the less amino acid differences we have in us compared to other organisms, whatever species it is, the closer we are on the evolutionary tree in relation to one another. Okay, so in other words, if we're looking here, it's just showing you guys how many differences there are between humans, monkeys, and gibbons, which if you look here, the sequences they're doing, they find like the same amount of differences, which is pretty crazy. But if we had like one difference between us and a monkey and then 5 differences between us and a frog, we would be in closer relation to the monkey compared to the frog. Okay, guys, that is it for chapter 3. So we are all done. Make sure you finish your study guides. Have a good rest of your day.